Hello everyone. Today we will talk about Ramachandran plot, and that was a remarkable contribution from Indian scientist in the area of world structural biology. The Ramachandran plot name was given on the name of the scientist who discovered it, Professor G.N. Ramachandran. Dr. G.N. Ramachandran was a great Indian physicist who was born in 1922 and survived up to 2001. And he was from University of Madras, where he served almost for a couple of decades as head of the Department of Physics and professor. Dr. G.N. Ramachandran, he was nominated 7 times for Nobel Prize, that also a record. And along with Ramachandran plot, he also discovered triple helical structure of collagen protein. We know that all the proteins, they have a unique shape, unique three-dimensional shape, means structure of the protein. And every protein, they also have a unique sequence of amino acids. So they vary in size from smallest almost 51 amino acid to even more than 26,000 number of amino acid in protein. The example of biggest protein known is cardiac muscle protein titin. And the shape of a protein is specific, and that's a requirement to its function also. The protein synthesis occurs by ribosomes in cytosol, and after translation is completed, or simultaneously protein sequence which is encoded from messenger RNA sequence that adopt a three-dimensional shape. And the major reason for adopting 3D shape for a protein, that is hydrophobic effect that we have discussed in earlier lecture. So because proteins have all the proteins which fold in three-dimensional space, they have hydrophobic amino acids, and because of those presence of those hydrophobic amino acids, after folding in cytosol, which is aqueous, proteins acquire a specific shape which is governed by their primary structure, and this specific shape is also responsible for that particular function of that protein. And three-dimensional structure of a protein is basically governed by main chain atoms. We know that in peptide bond formation, the main chain atoms CONH, C-alpha atoms of amino acids in a protein sequence, those are main chain atoms. So basically main chain atoms of a protein, they determine shape of that protein. That is, shape is also three-dimensional structure of the protein. The side chain of amino acids, they are responsible for particular function of the protein and also stabilization of three-dimensional structure of the protein. But as far as shape is concerned, that is majorly governed by main chain atoms of the protein, and that's why a protein folded shape is described by the path traced by its peptide backbone. So this is the backbone of amino acids which determine what kind of shape a protein is going to acquire after folding has occurred in cytosol. Now let's look at a protein structure. This is surface diagram of a protein. Basically this is protease from SARS coronavirus 2. And here convention wise you should remember that blue color represent nitrogen atoms, red oxygen, and here carbon could be anything, like here is given green color. So proteins are generally in nanometer dimensions, and if you look at they are all amino acids which are there in the protein. They form interactions, attractive interaction with other amino acid side chains, and that's how protein structures are stabilized. Now if you see carefully, this is one terminal here. This basically is N terminal, the first amino acid. This is C terminal, the last amino acid, and the protein chain goes like this, and 2 and 4th, and that's how a protein acquire a specific shape in three-dimensional space. And if you look at secondary structural elements in a protein like alpha helices, so the red color things, they represent alpha helices. So in alpha helices, like here, the protein chain is going in a spiral way. So when protein chain is going in a spiral way in alpha helix, it requires rotation of some of the bonds. Similarly, in case of beta strands also, the yellow color in the figure, the yellow color lines, they represent beta sheets. They are also not extended conformation. So like they are going in a zigzag fashion. So they also require rotation of the bonds. Or if you look at the surface loops, for example here, so protein chain is going like this, then is instead of going straight, it has to turn back in order to give a shape to the protein. So when such kind of bending is present, that require rotation of the bonds of the main chain. These structure represent only main chain atoms of the protein. Side chains are omitted here. So in this case, the rotation of these bonds, they are basically two types of bond. You call them Phi and Psi bonds, which are responsible for protein folding, so that protein can acquire a shape in three-dimensional space in order to perform a particular function. Phi and Psi, they are dihedral angles. They are also known as torsion angles or planar angles. So Phi and Psi are basically angle between two planes. For example, to define a torsion angle or dihedral angle, you require minimum 4 atoms. For example, if you have, say, 4 points A, B and C, D, and A and B, they are located in one plane, like shown here in violet color, and C, D atoms, they are forming another plane. The best example is if you look at a book or a file cover. So in this situation, the angle between these two planes, AB plane, this one, and CD plane, the green color. So the angle between them across the bond between B and C. So if you rotate this bond between B and C, these two planes, the position of these two planes can be changed with respect to each other. And the angle which varies between these two planes, that's called dihedral angle or torsion angle. Similarly, when you open a book or close a book, if you look at the pages of the book, so those pages, the positioning, relative positioning of the pages changes. The angle between those pages, that varies. It can be 0 degree if you close the book. If you completely open the book, the angle, the planar angle between those two pages, that is 180 degree. So that's how you can represent torsion angles. And Phi and Psi, they are basically nothing but they are dihedral angles which present in main chain of the protein. Before we talk about Phi Psi map, we need to look at the peptide bond. This also we have discussed earlier in some of the lecture in more detail. Peptide bonds are basically is a bond which is produced between two amino acid. Two amino acid react together, form a peptide bond. This is also called as amide bond, which is generated by a dehydration synthesis reaction. And this CONH bond, this joins different amino acids in structure of a protein. So, and this CONH bond is not basically a single bond like you have C and N here. It has acquired a partial double bond character due to resonance. So that partial double bond character gives rise to special properties to the peptide bond. Because of presence of this partial double bond, this peptide bond is become rigid and planar. This bond cannot be rotated. The bond between C and N in the peptide bond, this is peptide bond here. So C and N bond is not freely rotated because of partial double bond character, and the bond distance, the C N bond distance is 1.33 angstrom, which is shorter than a typical CN single bond and is longer than a double bond, CN double bond, which is 1.25 and 1.47. And this double bond character in peptide bond is 40%, and all the 6 atoms, they are planar. 6 atom means the C alpha atom, this is one amino acid C alpha atom, C oxygen, NH of another amino acid, and C alpha atom. So 1, 2, 3, 4, 5, 6, these 6 atoms, they are planar in nature. And of course it has got a dipole moment also. This we have discussed in earlier lecture about peptide bond. And the bond angle which represent between planes of carbonyl oxygen of peptide bond CO and NH, this is designated as omega angle. Means if CO and NH, they lies in opposite planes, the angle between them is 180 degree. This is called transconfiguration. So CO and NH, they are facing opposite side, and the peptide bond is transconfiguration. In more than 97% cases in proteins, this configuration is found, this more favorable. But in less than 3% cases, this CO and NH both can face same side also. That's called cis configuration, and the bond angle in cis configuration, also called as cis peptide bond, is 0 degree. So bond angle means the angle between plane of CO and plane of NH. So this is 0 degree in case of cis peptide bond, whereas it's 180 degree in case of transpeptide bond. But there is another problem in case of cis configuration. The side chain, in order to rotate carbonyl group, for example here towards the plane of NH, so this C alpha, this R group here should rotate in other side, and that's how this R group of these two consecutive amino acids which are forming cis peptide bond, they clash with each other, and because of this steric clash, cis peptide bond is not favorable. It is not found in very much in protein structures, but less than 3% cases cis peptide bond occurs in the protein. So in those situations, this amino acid should be something which is not having R group. If R group is there, it will clash with the R group of another amino acid, so cis peptide bond formation is not possible. But if you have amino acid like glycine, in case of glycine we know there is no side chain group, so glycine does not make steric clash with this amino acid. So that's why in most of the cases, whenever cis peptide bond is found in proteins, glycine is present as a partner of the peptide bond because it lack side chain. Now this peptide bond has limited rotation because it can be either 0 degree or 180 degrees, trans configuration or cis configuration. It has got a partial double bond character also. So for example, in this case is a dipeptide. There are two peptides. The plane of amide is shown with the blue color, and this bond CN bond is because it's a partial double bond, so it's not free to rotate. So in case of polypeptides, there are only two single bonds which are free to rotate. If you look at this chain, main chain atoms, this is C alpha of one amino acid, and NH is not shown. Then CO of the same amino acid, NH of another amino acid, this is C alpha of next amino acid, CO group and NH group, and this is C alpha of third amino acid. So in this situation, in the main chain, CN bond is not free to rotate. That's what we have seen. This omega angle we are calling that. So this cannot rotate, but the bond between C alpha and C and N and C alpha, they are single CC and CN bond, they are free to rotate. They can rotate, so they are designated as phi and psi. The angle between N and C alpha is called phi rotation because this is CN single bond is free to rotate. Similarly, the C alpha and C, this bond is called phi bond or phi angle, that's also free to rotate in case of protein structure. So for example, if you look at this dipeptide, this is the plane of a peptide bond, one peptide bond and another peptide bond. So in order to have a rotation of phi and psi, if you freeze this bond, this entire plane, and you try to rotate across this C alpha and N, this is called phi rotation. Similarly, if you freeze this structure and you try to rotate across C alpha and C bond, this is called psi rotation or psi bond. These phi and psi are the only flexible bonds which are free to rotate, and that's how they rotate in order to give a specific shape to the protein when protein folds. So these phi and psi angles, as we know, these are planar angles, and they are defined by 4 atoms of the main chain of polypeptide. For example, you have N and C alpha, N and C alpha bond, then C alpha, and this is C atom. The blue color is nitrogen atom. If you look at the plane of C alpha and N, they are in one plane, and then C and N, these are in another plane, and in order to change planar angle between these two planes, you need to rotate the C alpha and C bond, and when you rotate this bond, this angle between two planes, that can vary from minus 180 degree to plus 180 degree. For example, here if you fix C alpha and N bond and you try to rotate this bond between C alpha and C, if the rotation is going in clockwise direction, this is represented in plus sign. So for example, it can be plus 60 degree or plus 120 degree, and eventually when it's totally extended, in that case it will be plus 180 degree, and that's also equal to minus 180 degree. Similarly, if you rotate this angle in anti-clockwise direction, minus 60 degree, minus 120 degree, and minus 180 degree. So that's how plus and minus sign are given to phi and psi angles. So phi and psi angles are defined as being plus minus 180 degree when polypeptide backbone is fully extended. In order to have completely extended polypeptide chain, phi and psi angle should be plus 180 or minus 180 degree, that's the same thing. But in case of proteins, because protein acquire different shapes, specific shapes, these phi psi varies from theoretically varies from minus 180 degree to plus 180 degree. And many of the values of phi and psi combinations, they are prohibited by steric interference between atoms in the polypeptide backbone and the side chain group. And the conformation where phi and psi, the angle between phi and psi angles are 0 degree, the planar angle, those are also not favorable, they are not allowed because of steric clashes. G. N. Ramachandran, he took peptide models, and in a computer he tried to change values of phi and psi, and same time he observed what are the values of phi psi which are permitted in order so that it does not make any clash when you rotate phi and psi bonds, and he concluded in form of Ramachandran plot. So let's look at a model here, peptide model. This is C alpha atom of one amino acid, this is C O group, and red is oxygen, N nitrogen, this is hydrogen atom in white color. This dark color, this is C alpha, then you have another peptide bond, CN bond, and CO, and another C alpha. So in this situation, this green color is phi bond. This is between N and C alpha atom. This is phi bond. If we try to rotate phi bond, what happens? So you see here, this is moving anti-clockwise direction. So it can rotate, it can freely rotate the plane of peptide bond C and N, it can rotate across this N-C alpha bond. Similarly, if you look at the psi bond is C alpha and C, this another one, and if you rotate that, it moves like this, clockwise direction and anti-clockwise direction. So this experiment was done by G. N. Ramachandran and his colleagues. He changed phi and psi values of peptide models, and then he made some observations. He also considered all the atoms as like hard balls which cannot overlap each other's space. Two atoms cannot come too close, otherwise they will make steric clash. Now let's look at the clashes. If you rotate these phi and psi, theoretically phi and psi can have any combination from minus 180 degree to plus 180 degree. But practically that does not happen, because in computer models, if you rotate them, some of the values of phi and psi which are not allowed due to steric clashes. Like for example here, this is the situation where there is no phi psi angles they are all allowed, there is no clash. But if you change them, this yellow color thing which also turn into red, it shows steric clash. So for example is going away, so here it keep going distant from atoms, and that also it keep making clashes. For example, at this orientation, this values of phi and psi, if you see this central amino acid, this is R group, methyl group, in fact this is alanine. This R group of alanine is become too close to this oxygen atom. That's what it is shown in red and yellow color. So that make steric clash here. It means this value of phi and psi is not allowed in this peptide. So it need to rotate more. So for example here there is no clash. So this is most favorable values of phi and psi which are allowed, which do not make any clash, steric clash between atoms. So that's what was done by Ramachandran. Then he rotated all the values, all the combination of phi and psi he looked upon. Similarly, for example, if you change here this NH atom, it become too close to the R group of this central amino acid. So that what was done by G.N. Ramachandran and his colleagues, and eventually he gave his postulates which are called Ramachandran plot. So that's how Ramachandran used computer models of small peptides. He systematically varied phi and psi of the peptides, and eventually he presented his information in form of phi and psi plot. For each conformation, while varying phi and psi values, the structure was examined for close contact. If in some values of phi and psi, the structure made close contact with some other atom, that was discarded. It was termed as disallowed conformation of peptide. And all the atoms, they were treated as hard sphere, and the dimension of the sphere was corresponding to the van der Waals radii of those atoms. And therefore phi and psi angles which cause spheres to collide, which is responsible for steric clashes, those were termed as disallowed conformation of the peptide backbone. And he also found that only 10% of phi psi combinations are generally observed for protein. Although theoretically phi and psi can vary from minus 180 degree to plus 180 degree, but in actual situation, when he was doing calculations using computers, he found that there are only 10% combinations of phi and psi which are actually allowed. Rest other combinations, they created steric clashes between atoms, and that's why they were called disallowed conformation of the peptides and protein. The interference in rotation of phi and psi was caused by spatial arrangement of atoms within molecules. And he also considered, because atoms cannot overlap, two atoms cannot come too close, otherwise their electronic cloud will repel each other. So considering all these conformation which were allowed and disallowed, Ramachandran postulated a plot between phi and psi for peptides, and the computed angles which were sterically allowed, they fall into certain region of the plot that he observed. So on the x-axis he took phi from minus 180 degree, 0 to plus 180 degree. Similarly on y-axis also he plotted psi, value of psi from minus 180, 0 to plus 180 degree. And he found out that only there are some regions of this plot which are occupied by allowed phi and psi combinations. For example, in case of a beta sheet, they fall into this region. Similarly, right-handed alpha helix, they fall here, and left-handed alpha helix on the right side here. And the red color here represent most allowed regions, means those phi and psi angle which fall into red area. The red color regions, they are most favorable. They did not created any steric interference between the atoms. So they were called most allowed phi and psi combinations, or most allowed regions of the Ramachandran plot. Similarly, you have yellow region also. The yellow region is represents additionally allowed region. Additionally allowed means this is not most allowed, but this also can occur because the steric clashes are less in case of these yellow regions, these combinations. And most of the area is basically white. White means those phi psi combinations, those conformations, those value of phi and psi, they are prohibited, because if amino acid acquire any phi psi combination which lies into white area, that is not allowed. It create steric clashes between the amino acid atoms. So this is called Ramachandran plot. And he also observed that only beta position influences the rotation. Beta position means if you have, say, main chain going like this, and you have side chain. In the main chain you have side group. For example, this is carbon atom in side group. In this case, then you have another carbon, and so on, depending on the length of the or nature of the amino acid. This beta position which influence more the rotation of phi and psi. Phi psi are basically across C alpha atom, the rotation of those bonds. That's what we have discussed about. So this C beta atom is the one which is in close proximity to these main chain atoms, and that's why the C beta atom that influence rotation of these phi and psi angles most. In case of any amino acid where there is no C beta, like in case of glycine, that does not influence rotation of phi and psi. That's why glycine, in case of glycine, rotation is free. Glycine can occupy complete area of the phi psi plot, Ramachandran plot, because in case of glycine, side chain is not there. There is no C beta atom, so the rotation is much more free in case of glycine due to missing side chain. So C beta position influence the rotation most. And in fact, if there is any beta branch amino acid, beta branch amino acid means you have main chain and this is C beta atom, and from C beta if you have branching like this, for example in case of valine or in case of isoleucine, these branching atoms, they are close to the main chain. So branched amino acid, they impose more restraints on rotation of phi and psi angles. So that's why even isoleucine and valine, they occupy lesser area of the Ramachandran plot, because in that case, due to beta branching, that impose more restriction on the freedom of phi and psi rotations. So glycine has no side chain, so it can occupy significant area of the Ramachandran plot. Most of the conformations from minus 180 to plus 180 degree of phi psi, they are permitted in glycine, and all residues excluding proline and beta branched, they occupy similar area in the energy map. For example, if you have, say, lysine has a long side chain, or you say about arginine or alanine, because in all these cases you have a beta position is there. For example, in case of alanine, you have methyl group here, and it can further extend it to another methyl group, and so on in case of other amino acids. So because the rotation of the bonds is influenced by only C beta position, so all the amino acids except proline and beta branch, they occupy similar area, because length of the side chain doesn't matter. If you talk about tryptophan or you talk about alanine, tryptophan is very bulky side chain, but the effect on Phi Psi rotation is due to C beta atom only, and C beta atom is present in all the amino acid except glycine or proline. So in those cases where no beta branching is there and proline, all the amino acid they occupy similar area in the Ramachandran plot. And of course glycine is a great exception because it can occupy maximum space due to missing side chain of the glycine amino acid. It's not only main chain but side chain of amino acid also, they have a carbon-carbon single bond and they are free to rotate. For example, here in case of tyrosine, if you look at this tyrosine, this is phenyl group and oxygen red, so it can occupy so many conformations because these bonds also can rotate, and the rotation of these side chain atoms, this is called chi angles, chi 1, chi 2 and so on, but it does not influence much about the rotation of phi and psi because phi psi are actually main chain angles. So this is an example of Ramachandran plot. Here you see that these dot lines, silver dot lines, they represent amino acids, combination of phi and psi for amino acids in a protein, and in an ideal situation, in a best structure of protein, all the amino acids should fall into the red area. Red area means which is most favorable, most allowed combination of phi and psi for a protein. And there should be no amino acid in area which is disallowed, like here. Even secondary structural elements, alpha helix and beta strand, they have a specific area of Ramachandran plot to occupy. In case of beta strands, in case of alpha helix, the value is minus 57 of phi and minus 47 for psi. So this is the area which is occupied by alpha helix. Similarly, beta conformation, in case of beta conformation, minus 139 value of phi somewhere here, and it will go up to here. So this is the area which is occupied by beta sheets. Even beta turns, the surface turns, loops, they also have a specific rotation value of phi and psi of amino acids in order to acquire that structure, that shape in protein. Ramachandran plot was given in 1960s, in 1963 in fact, and since then it is being used in validation of protein structures. When protein structure is determined by X-ray crystallography or NMR or any other method, in order to check their geometry, in order to check their conformation is correct or not, Ramachandran plot is used. For example, this is a poor example of Ramachandran plot of protein structure, where we can see that many amino acids they lie into white area which is disallowed. So if you have this kind of structure, it means the structure is not correct. Ideally, there should not be no amino acid in disallowed region. This is also called outlier. In outlier region, there should not be no amino acid. Most of the amino acids should lie into the red areas, or if not red, some amino acid can lie into this gray area or very little in yellow area. So in fact, experimentally, there are 4 regions of Ramachandran plot. The red, which is most allowed, and this is shown here with brown, this is additionally allowed regions, and the yellow region, this is generously allowed region, and of course this light color is disallowed region. So ideally there should not be any amino acid in a structure of a protein which has amino acid Phi Psi values lying into disallowed regions. So maximum more than 90% amino acid should be in the red area. Some amino acid may present into the brown area, which is additional allowed region, and there should be least number of amino acid in yellow area, which is generously allowed region. And ideally no amino acid should have Phi Psi combination in disallowed area of the Ramachandran plot. So Ramachandran plot since 1960s is still being used in validating structures of proteins to check whether their conformation of amino acids they are allowed or disallowed. So this is another example of a good Ramachandran plot where we can see that most of the amino acid they are falling into red region, which is most allowed, and little number they are into additional allowed regions also, that's permitted. And there should not be, there is no amino acid which is lying into white region, that is disallowed. If you see glycine, these are glycine amino acid which are present in white area. So glycine can occupy anywhere in fact. So when Ramachandran calculations are made, glycine is excluded because glycine is free to rotate. Phi and psi for glycine can be anywhere from -180 to +180. So that's why glycine is not considered into Ramachandran plot.